Hello, my name is Cory Gardner, and I'm here today to present research and results for the CAMDA Metagenomic Challenge on behalf of myself and my teammates at St. Louis University, Sadia Ahmad and Dr. Ted Ahn. In this presentation, I'll begin with a very brief discussion of metagenomics in general and define the CAMDA Metasub Challenge. I'd then like to discuss the data sets provided for the challenge and some of the challenges that we ran into in their analysis. I'll then discuss our research questions that we posed for the challenge and the methods that we undertook to answer them. And finally, we'll unveil our mystery sample predictions and look at possible direction for future research. Metagenomics has been of increasing interest to researchers for a great many reasons, including its connection to human health and disease, its promise as a method for biosurveillance and tracking pathogens, and its ability to allow researchers to study microbial communities that can't be cultured in a laboratory. But along with the promises it gives are also a great many challenges. For us, they mostly revolved around the size of the data sets themselves. The data provided by CAMDA Metasub for this particular challenge took up nearly 6 terabytes of disk space uncompressed and consisted of over 18 billion reads. So along with such massive data sets come massive requirements for computational resources and a number of challenges in quantifying and analyzing the results. For us, we realized this pretty early on when we knew that we had approximately 2 months to complete all of the research for this conference, and it took us nearly 5 days just to download the data. We realized we had to be very careful in our methods and choose profiling and analysis methods that could be completed in a reasonable amount of time with the computational resources that we had available. So the data consists of two sets: a primary training set that has 1,065 paired-in samples from 23 cities, and this data set is labeled with the city of origin. The second set, or the mystery data, consists of 121 samples, but the mystery is that it doesn't tell us the city of origin. That's what we have to predict. So something that I didn't fully understand early on was the difficulty in just determining which of the mystery samples are actually represented by samples from the same city in the training data. It seemed to be one of the more difficult decisions in my opinion, and we'll discuss that more in a bit. So previous research from St. Louis University for the CAMDA Metasub Challenge determined that sample prediction remained really high even when only half of the whole genome sequencing data was used. So we decided to extend upon this research and consider the effect of reduced representation at various levels on mystery sample prediction. So our research questions. What is the impact of reduced representation of training data on prediction accuracy? How can we incorporate factors in the metadata for mystery sample prediction? Can we predict the geographic origin of a metagenomic sample even if no reference samples from that location are known? How can we determine what metadata factors are most predictive of sample origin? And how can we even determine which mystery samples are represented in the training data? So for the first question, reduced representation. We found that randomly sampling 6 terabytes of FASTQ files 9 times for 10% to 90%, 10% intervals was computationally prohibitive with the resources we had available. So instead, we developed a method to simulate data sets reduced representation data sets from the operational taxonomic unit tables that we generated from a full Kraken profiling of the WGS data. To do this, the relative species abundances of each sample were determined by dividing each species count by the total count of the species within the sample. And then the relative abundances were reduced by the desired representation level. And finally, a normal distribution centered at each of these values was generated, and a value is randomly selected within that range. Benchmarking the accuracy of predictions based on our reduced representation data set was a bit of a challenge in itself. We took two methods. First, we used 70-30 test trained data sets with random forest. And then after the true labels of the mystery data were released, the cities from that were common to both sets, the training and the mystery sets, were selected. We did reduced representation on the training sets, used that data to build random forest models to predict the mystery sample origins. So we found that in both cases, sample prediction remained consistent and rather high at all of the representation levels we tested. The 70-30 data performed slightly higher. We thought perhaps that may be because the history samples were from a different year, and microbial communities of the cities may have changed slightly. But in any case, we also realized that our method of the reduced representation data sets was only possible because of the massive size of the data sets. So here were our results from the reduced representation on prediction accuracy using the 70-30 split. As you can see, our prediction remained at 90%, whether we were looking at 10% reduced representation or the full data set. So it seems that with such massive data sets, the reduced representation of the data doesn't have much of an effect on the accuracy of the model. So our methods for analysis. First, we did FastQC. We found that both the training and the mystery sets had very high quality, a Phred score of at least 35. But we interleaved the paired in files. We did adapter removal with bbduke and then did human contaminant removal against the HG19 human genome with bbmap. We then used linear discriminant analysis to explore some of these sources of variation and how different grouping factors would show better clustering and possibly be more promising for prediction on other factor levels, which we'll discuss again in a moment. And then we tried different approaches for taxonomic profiling. First, we used Kraken 2 and Bracken for abundance estimation. And we tried Metaflan 3. And at least for us, in this particular case, with this particular data set, we found that the Kraken-Bracken combo was far more sensitive than the Metaflan 3 and identified far more of the species in our samples. We then used random forest for classification, and then we will discuss our method for analyzing our results. So first, linear discriminant analysis on the basis of city shows clustering among the cities, but there's a lot of overlap as well. And the results weren't always perfect. But we decided to see what would happen if we grouped the cities in various ways. And we found that coastal distance, like the distance from the nearest coast, elevation, the hemisphere, and the continent showed much cleaner clustering, and we felt showed promise for adding to our prediction accuracy. So the question was how we incorporated that information. So what we did was we initially took a we extracted the vote counts from random forest classification. And those samples that had results that were 3 standard deviations or above, we assumed as correctly classified and eliminated from further analysis. Samples that did not have a clear winner, as it were, we then classified on the basis of the other factors that we, such as distance from the coast, elevation, continent, and hemisphere, and we made predictions based on that. So for the secondary predictions, of course, like we couldn't say predict precisely what city they came from, but we hope to say uh, something about the type of environment in which we believe they came. So as you can see here, the grayed out samples are those that we predicted on the first round and eliminated from further analysis. And among those, we were fairly pleased with the results. We predicted 61 cities um, in the first round and assumed that they were represented in the training set. And in fact, a little over 80% of them were, and they were classified accurately. On the other factors, um, the majority of which we had right on all levels. Those that we missed were generally missed on one classification, and the classification that seemed to perform the most poorly for us was elevation. In future research, we would be very interested in identifying the other factors provided in the metadata, of which there were over 1,000, and which would be most predictive of sample origin. So, for example, if we weren't able to say that a sample was from Bogota, we were at least able to say something about the elevation of its geographical origin, what hemisphere it was in, what continent it was on, and um, and we were happy with those results. But we think that we could incorporate the other factors and narrow it down even more. So I'd like to say thank you to the organizers and hosts of this challenge, and we hope to do future research on this project. We really enjoyed it. It was tremendous uh, learning opportunity, and um, and I'd also like to thank my fantastic help of my research partner Sadia Ahmad and Dr. Tedon. Thank you.